Technobotany by Zura. Prime Editing Guide RNA, pegRNA, Design, Principles and Strategies. In this video, I will be exploring the crucial aspects of pegRNA design for prime editing, a revolutionary genome editing technique. We'll go through the fundamental concepts, design principles, and practical considerations to optimize your editing efficiency. What is prime editing? Prime editing is a search and replace genome editing method. It uses a Cas9 nickase fused to a reverse transcriptase and a pegRNA that encodes both the target site and the intended edit, making it a powerful tool in both basic research and therapeutic development. Prime editing is versatile and capable of performing all 12 possible base-to-base substitutions, as well as precise insertions and deletions. What is pegRNA and what is its anatomy? A typical pegRNA is a specially engineered RNA molecule that directs the editing process. It includes a spacer to target the genome, a primer binding site, PBS, to initiate reverse transcription, and a reverse transcriptase template, RTT, that carries the edit. The scaffold ensures Cas9 binding. Designing pegRNAs requires careful tuning. When designing the spacer sequence for Cas9 or Cas12a, a few key principles guide the process. First, choosing a target site. Select a unique genomic region that's relevant to the intended edit. Exonic regions are preferred when the goal is gene disruption or correction. It's best to avoid repetitive sequences and regions with excessively high GC content, which can reduce targeting efficiency. Second, PAM proximity. The spacer must be located near a PAM sequence that matches the requirements of the chosen nuclease, such as NGG for SpCas9 or TTTV for Cas12a. Third, off-target considerations. Use prediction tools like CRISPR or ChopChop to identify potential off-target effects. Spacers should have minimal homology to other genomic sites, especially in the seed region, the first 10 to 12 nucleotides adjacent to the PAM. For improved specificity, consider high-fidelity Cas variants, such as SpCas9HF1 or ESpCas9. Designing the RT template for pegRNA. The RT template is the heart of the pegRNA. It encodes the desired edit and guides reverse transcription. First, incorporating desired mutations. The RT template must encode the exact nucleotide changes, whether substitution, insertion, or deletion. It's often helpful to include silent mutations to disrupt the PAM site or to reduce mismatch repair. For maximum editing efficiency, ensure the mutation is positioned close to the nick site. Second, length considerations. RT templates typically range from 10 to 35 nucleotides. A good starting point is around 16 nucleotides, adjusting as needed based on the complexity of the edit. While longer templates can improve the efficiency of larger edits, they also increase the risk of forming secondary structures. Third, ensuring proper homology. The template must maintain strong homology with the target strand flanking the edit. Avoid starting the RT template with cytosine, as this can interfere with the gRNA scaffold. Tools like PegFinder or PE-Designer can be used to optimize both the sequence and structure of the RTT. Designing the primer binding site. The primer binding site is essential for initiating reverse transcription in prime editing. First, length considerations. The optimal PBS length ranges from 8 to 17 nucleotides, with around 13 nucleotides as a good starting point. Shorter PBS sequences may reduce binding strength, while longer ones can increase the risk of off-target priming. Second, GC content importance. When designing the PBS region of a pegRNA, an ideal GC content falls within the range of 40 to 60%. This balance ensures optimal hybridization between the RNA and the DNA target. A higher GC content can enhance duplex stability due to stronger hydrogen bonding. However, it also increases the likelihood of forming unwanted secondary structures, which may impair function. Therefore, it is advisable to avoid sequences that are either too GC-rich or too at-rich, as extreme compositions can compromise the efficiency and reliability of the system. Third, stability and binding efficiency. The primer binding site must anneal precisely to the 3' end of the nicked DNA strand, as efficient priming is critical for initiating reverse transcription. Avoid sequences prone to hairpin loops or self-complementarity. Tools such as pegFinder or PrimeDesign can be used to evaluate and refine PBS variants. Several bioinformatics tools simplify pegRNA design by automating spacer selection, PBS length, and RT template prediction. PrimeDesign, developed by the Lu Lab, is a comprehensive platform tailored specifically for prime editing guide design, offering automated generation of pegRNAs and secondary nicking sgRNAs. pegDesigner, part of the RGEN tools suite, provides a user-friendly interface to design pegRNAs with customizable parameters and on-target/off-target predictions. Benchling CRISPR tools offer integrated workflows for pegRNA design, molecular cloning, and project management, making it ideal for collaborative research environments. CRISPR RGEN-Cas is another versatile tool, supporting pegRNA construction alongside various CRISPR nucleases and editing systems. I hope you enjoyed this video.